we can get started first. People will join us later. Welcome to everyone to our bioinformatics and genomics seminar today. We are very glad to invite Dr. Sagi Shapira from Columbia University to talk about their Nature Medicine paper. And usually we have bi-weekly seminars, and for last week we have one extra topics. It's regarding also, I think that's the last topic regarding the COVID-19. And if you are interested, you are welcome to join us. And in March, we have several topics about PRS score and the copy number variation. And in April, we have topics about single-cell RNA-seq. So if you are interested to any of this project or topics, you're welcome to join us. And for what previous seminars, if you miss any previous seminars, and if you think you are interested in that, you are welcome to come to this page and click the link. And what previous seminars was recorded in YouTube. With that, I will turn it over to Dr. Al Ozono from BCH. So who will co-host this meeting with me and also introduce our speaker. Thank you, Al.
Thank you so much, Lang, and hello, everybody. For those that don't know me, I'm the Associate Director in the Precision Vaccines Program based in the Division of Infectious Diseases. And I'm very pleased now to be introducing Sagi Shapira for the talk today. Maybe I'll just say very briefly, just give everybody an idea about Sagi's background. He completed an MPH in infectious disease epi at Yale, a PhD at UPenn in immunology and parasitology, postdoc at Harvard Broad, and now is assistant professor at Columbia, where his lab focuses on identifying biological principles at the interface of virology and innate immunity. His lab is multidisciplinary, brings together tools and concepts to engage in hypothesis-driven systems biology, as he describes it, experimental and computational methods to make non-incremental advances, studying the interface of pathogen-host interactions and human physiology. Sagi and I had a chance to chat briefly before this seminar, and clearly have some common interests. So I'm very much looking forward to his talk today, immune complement and coagulation dysfunction in adverse outcomes of SARS-CoV-2 infection. This is work that has been published in Nature Medicine, and I hope you'll all join me in welcoming our speaker for what I'm sure is going to be a terrific talk. So with that brief introduction, Sagi, we're really looking forward to what you have to say, and the floor is yours.
Thanks so much, Al, and thanks for the invitation. And I'm gonna let's see if I first share. I imagine everybody can see this. Yes. Okay, brilliant. So I'm gonna try. And I was asked to cover the paper that we published, because it's almost a year now, which is hard to believe. But I thought that it would be also useful for people to hear sort of what drove sort of the discovery that we made, the papers that sort of led up to it, and maybe at some point even talk about some unpublished work. As Al suggested, the work in my lab, we focus on trying to understand the interface of pathogens and hosts. In particular, we focus on viruses, but we take sort of a combination of experimental and computational methods to learn what the circuitry is at the interface of those virus-host interactions and how cells make decisions, how cells decide what genes to turn on and off, and sort of learn about what are the key regulators of both innate and adaptive responses. And so I will focus today on the computational side of the work. But before I go too far into things, I want to just sort of highlight a few people that made the work possible. Gorka was a postdoc in my lab. He now has his own group at Albert Einstein. I'll be talking a lot about his work. Barry Honig is a close collaborator in the Department of Systems Biology at Columbia. And a lot of the discoveries were motivated by this close collaboration. And then the paper that I was invited to sort of talk about is one that was a collaboration between myself, Nick Tetnetti, and Chris Mason, and it's a collaboration that's ongoing and has been quite fruitful and illuminating a lot of very key drivers of SARS-CoV-2 infection and downstream disease. So when I start my talks, I always have this slide because, and perhaps with this audience, it's not that critical, but I think it illustrates really well the intimate relationship that pathogens have with their hosts. So, pathogens come in different flavors. There are eukaryotic parasites. Trypanosomes interact directly with red blood cells. Toxoplasma is a parasite that is very near and dear to my heart. I did my PhD on Toxoplasma, and you see here it interacts directly with nuclei of cells that it infects, and you don't see on this picture mitochondria and ER that it actually uses to get nutrients. There are multicellular parasites like hookworms and Bordetella that sort of interact with tissues to get nutrients. And of course, there are bacterial pathogens that use type 3 secretion to introduce toxins that manipulate cellular responses. And viruses, of course, which are particularly timely today, are interesting for various reasons. Of course, they're public health concerns, but viruses offer something that the other more complex pathogens don't, and that is that they have relatively very small genomes and proteomes, and they're often experimental tools in genetics that allow us to manipulate the pathogen and allow us to take sort of a more holistic view of how these interactions are dictated both by viral proteins and viral encoded factors, as well as cellular sort of responses. Now, I want to point out before I go further, I want to point out one key thing that I think is always important to keep in mind, and that is that as experimentalists, we live along this sort of axis. The greater the number of perturbations that we impose on a system, the fewer number of measurements that we can monitor, and vice versa. The greater number of measurements, the fewer number of perturbations. And so we sort of live along this axis. And what we try to do in my lab is we try to use what I hope are clever and interesting experimental methods, but also computational tools to try and push this envelope. And that's sort of what I think of as hypothesis-driven systems biology, where we allow biological questions to sort of drive the experimental and computational methods that come downstream. Now, as Al pointed out, I did my postdoc in Boston near Hakohen's lab together with Aviv Regev. And one of the things that I was interested in is learning about pathogen host interactions. And as I said, we were interested in viruses at the time. And what we did was we applied sort of an approach that would allow biology to guide discovery of key regulators in influenza pathogenesis. So this is published, so I won't go into details. But what we know is that when we examine what are the physical interactions, the protein-protein interactions between pathogen and host, and the transcriptional programs that are induced, that can help guide discovery of really key regulators of that infectious life cycle. And that can tell us about what cellular factors are important for completing the viral life cycle, but also what cellular factors are important for the response to infection. And not just this work, but there's a very long literature from many labs that have shown that discovery of these cognate physical interactions is really critical in guiding discovery of those cellular factors that control infection. Now, imagine if you could, rather than looking at one virus at a time, you could build this interaction network for all viruses, or perhaps even all pathogens. Now, there are, of course, experimental methods like use to hybrid or mass spec, or very targeted approaches to discover protein-protein interactions or complexes of interactions. But if one wants to develop this sort of cartography or architecture across all pathogens, then those approaches, experimental approaches, simply don't have the throughput to be able to examine sort of more broadly. So that's where this algorithm called phipster was born. Phipster is a derivative of another algorithm called PREPI, PREPPI. PREPI was developed by Barry Honig, who I mentioned at the first slide. PREPI, what PREPI does is it uses things like gene expression, evolutionary conservation, annotation of protein functions, the idea that if things are expressed at the same time and evolve together or have similar functions, they're more likely to interact. But what PREPI does that's really special is that it uses protein structure to evaluate whether pairs of proteins are likely to interact or not. Now, PREPI was published splashy paper in Nature, and they applied it to human, and they showed that it works. It's very accurate, and that was kind of where it sat. I joined Columbia now almost 9 years ago, and my idea was, well, if PREPI does such a good job with human protein-protein interactions, why not do this for viruses? Turns out that it's not that simple. With viruses, we don't have things, non-structural cues like function or when proteins are expressed or evolutionary conservation. For the vast majority of viruses or pathogens, really, we know much beyond genome sequence. And so we can only rely on structural information, protein structure information. So we developed this algorithm that takes advantage exclusively protein structure and structure modeling. If people are interested, again, this is published, so I won't go into too much detail. And if people are interested, I can go into further detail. But what we do essentially is we take viral proteomes, and we build atomic structure models of the proteins that they encode. And then we evaluate pairwise whether a pair of interactions is compatible or not. And with that, we build sort of a cartography of pan-virus human interactions. So we applied this to all human-affecting viruses. there are 1,000 human-affecting viruses. They're represented by about 13,000 proteins. And so we have a matrix of the 13,000 viral proteins versus the 20,000 human proteome. And in each cell, we have a sort of likelihood that a pair of proteins will interact or not. And we found about 280 or so thousand novel protein-protein interactions. I'll skip this. And as an experimentalist, there's something very, very satisfying about using 40 million compute hours to end up with some result, but you want to know that that result is actually meaningful. And so we were able to show that we rediscover known interactions, and we actually went a step further, and we took about, I think, maybe 80 novel pairwise interactions. We evaluated them and showed that we have a very high true positive and true negative rate. Really remarkable, as far as I was concerned. Now, the thing is that, as I said, knowledge of these protein-protein interactions, or the fantasy, was that it will help guide discovery of key fundamental principles in pathogen-host interactions, but also allow us to discover key determinants of cellular responses to infection. And I won't go into too much detail, but because again, it's published, but suffice to say, we were able to do at least some of that, and add a layer of complexity to our understanding of virus host interactions. Okay, so another way that pathogens can manipulate the cells that they infect, and viruses are sort of specialists at this, is through mimicry. And mimicry is something that is very well described in ecology. And of course, everybody's seen these pictures of butterflies that sort of look like the tree bark that they sit on. And there are these fish that look like their environment. They have like eyes on their tails, and there are these worms that mimic snakes to scare their predators. And viruses are particularly good at this, and DNA viruses are particularly good at this, because DNA viruses can pick up genes from their host through horizontal gene transfer. Now, discovery of mimics has been hampered by the fact that viruses are genetically highly diverse, and the methods available for discovering mimics and protein mimics rely heavily on sequence-based sort of searches. Now, since the sequence divergence is so high, the examples in literature of virus-encoded mimics are limited to these DNA viruses where the gene has been picked up by the virus, and sequence similarity is relatively high, 30–40% sequence similarity. We took an approach that sort of relied on what I described before, where we build atomic structure models of proteins and ask whether we can find proteins that look similar to a query protein. So the idea is essentially, rather than doing sequence alignment, we build a structure model of a protein sequence, and then we use a computational method that aligns structures in 3 dimensions. Okay. And we can evaluate how similar a pair of structures is, and I'll show you a couple of examples to give you a sense for what it looks like. But what we did was we built these structure models for proteins for all viruses that have been described, and ask whether the structure space that's used by viruses is dictated, at least in part, by the host that they infect. Okay, so here's an example. So we look at all phages. We build structure models for all phage proteins that have been described, and we ask whether those proteins are more similar in structure to various hosts. And perhaps not surprising, the structure language that phages use is most similar to E. coli. Makes sense, they infect bacteria, and not very similar to yeast or human. Now, we can take it one step further, and we can look across all hosts and viruses in sort of Earth's virome. And again, perhaps not surprising, bacterial viruses look like bacteria, plant-infecting viruses look like plant, and viruses that infect vertebrates look like vertebrates. So it's not entirely surprising, but it's nice to sort of demonstrate. But what's really remarkable is that when you look at viruses, arboviruses, these are viruses that infect people but are transmitted by insects. They use a structure language that is similar to both mosquito or insect and human. So it suggests that they encode proteins that are structurally promiscuous and that allow them to complete their life cycle in both their insect and human hosts. Really, it's awesome. Really, really awesome. So what does it mean for people? Well, turns out that, as I said, viruses are very good at this, and they explore structure space very efficiently because they mutate so rapidly. But what's very interesting is that RNA viruses, which their genomes are on the small end of the scale, they encode for a relatively small number of proteins. RNA viruses are sort of specialists. They mimic many host proteins. And among RNA viruses, coronaviruses are particularly good at this. So coronaviruses are RNA viruses. They, as I imagine everyone knows by now, and they encode, I think, something like anywhere between 150 or 160 mimics. Their proteome is only 30 proteins, and they're able to mimic about 150 human proteins. And there are a few things here that some folks will, the aficionados will recognize. So just to orient you, these are the various coronavirus proteins for which we find mimics, for which we find that they mimic human protein. This is the sequence identity. The viral proteins are in red, and blue are the human proteins. And just to give you a sense, you know, when we talk about mimicry here, we look at proteins that look virtually identical, but the sequence identity is often around 5% or less. Really, really striking. Here's a couple of proteins, DDX58. For those of you who work on innate immunity, DDX58 is RIG-I. This is the cytosolic sensor for viral RNA. DDX3X is a modifier of RIG-I dependent nucleic acid sensing. In fact, I believe it also regulates DNA sensing. PARPs, these regulate signaling downstream of the interferon receptor. And of course, type I interferons are critical for controlling antiviral responses. As their name suggests, they interfere with the viral life cycles. TMPRSS1, TMPRSS2, of course, has been in the news a lot for its role in coronavirus infection. And the one set of proteins that really stuck out were members of complement and coagulation pathways. And this is where I'll sort of go into more detail next. But this is where the paper sort of ended. We were ready to submit, and in fact, we submitted it as it is, and then the pandemic started, and for those who follow the coronavirus literature, we've known for a long time that patients who are infected with SARS coronaviruses develop sort of coagulopathies and sort of complement and coagulation mediated disease. And so this was quite interesting to find not just complement 2, but many complement and coagulation factors being mimicked by coronavirus. So we decided to take a sort of an approach, which is, as I started saying, the whole premise of this work is to allow the biology to guide discovery of key factors, cellular or physiological factors that drive disease. Now, some of these factors may influence the viral life cycle, some may influence immune response, and some may influence the pathogenesis of disease. So we start off with this database of structure encoded mimics encoded by coronavirus. At the time when we started this project, we were unfortunate enough that in New York, at least at Columbia, we had something like 11 or 12,000 patients. This was in, I would guess, March or April. By March or April, we had about 11,000 patients that were SARS-CoV-2 positive and that were seen at the hospital. That number allows us to do some, interesting informatics. It's a large enough number that we can look at the contributions of various clinical histories in the outcome of disease. And something I didn't remember too much about complement, but something I do remember from when I took immunology is that complement is associated with macular degeneration. One of the drivers of macular degeneration is a hypercomplement phenotype. And in fact, even patients that do not have complement dysfunction, patients that have macular degeneration that do not have complement dysfunction, the disease is associated with hypercomplement activation. And we can talk a little bit more about this towards the end of the discussion. So we took a relatively naive approach. We simply asked whether a history of macular degeneration or some coagulopathies or some of the other risk factors that were had already been described were associated with increased risk with either mortality or morbidity. So morbidity was essentially defined as intubation. Mortality, of course, is people who succumb to disease. And we stratified patients based on age, sex, history of macular degeneration, coronary artery disease, coagulation, and you guys can read the rest. And of course, we rediscovered known risk factors and found that history of macular degeneration was indeed associated with far greater susceptibility to both intubation and mortality, and history of coagulopathies were also associated with the same. We can actually measure what is the risk associated with having a history of macular degeneration or one of the other risk factors. And you can see that even when we correct for age and sex, that's in red, versus gray, which is uncorrected, we still see a very significant contribution to both morbidity and mortality associated with SARS-CoV-2 infection. And actually, something that I should have mentioned here, we also identified patients that have complement deficiencies. So these are patients that are otherwise very immunocompromised. At the time, we had 4 patients. We're now, I think, up to 11 or 12. None of them required either intubation or died as a result of their infection, suggesting that, in fact, that really complement activation participates, at least in some way, in the sort of COVID outcomes. We, in addition to looking at sort of clinical associations with of disease, we also looked at the engagement of complement and coagulation pathways in nasal swabs of infected patients. At the time, we had, I believe, this was a cohort of about 600. We're now going to have a far broader cohort. But what you can see is that infection, perhaps not surprising, engages sort of type 1 interferon responses and very generic inflammatory responses, but the complement and coagulation pathways, these colored circles, are highly induced in a virus-dependent manner in infected patients. For the aficionados, you can look at what the genes are that are involved in these pathways, but what I think is most remarkable is that we could go from this prediction of complement being associated and being engaged during SARS-CoV-2 infection to actually asking whether mutations and polymorphisms in complement-associated factors is or is not associated with severe disease. So at the time, there were a couple of cohorts whose data was released in the UK Biobank. So for those of you who are not familiar, and I imagine this audience all is, UK Biobank is following tens, if not a couple of 100,000 patients that have been fully sequenced or genotyped in one way or another. And at the time, there were, I think, roughly 1,100 patients that had been genotyped who had been hospitalized with SARS-CoV-2. And what we could do is actually, rather than doing a GWAS, we could do a targeted screen to ask whether mutations in these factors are associated with disease. And indeed, we found a number of them, and they repeated across 2 different cohorts, and suggested that indeed complement and coagulation polymorphisms may contribute, at least in part, to severity of disease. This slide is a bit old, but one of the things that this has sort of motivated is the use of small molecule inhibitors that were actually initially developed for other complement-associated diseases, now being used in COVID patients. And it's actually, the results are quite remarkable. For many of the patients, the severity of disease has really been turned around, and many patients have been completely cured. So it's quite promising. These trials are now, I believe, going to phase 3, and certainly hope that they can alleviate at least some of the morbidity and mortality associated with SARS-CoV-2 infection. So I'm going to stop there and take some questions. I wanted to leave time for discussion and then sort of see where that takes us. We can always go into more details on questions and sort of things that are ongoing and that we've done. And let's see where the discussion takes us. So, any questions?
Thanks again. If it's helpful, I'll try to moderate, and maybe the thing to do is use, if we have a raise hand function, Zoom here or something like that, if that's helpful, or please, folks, just feel free to jump in.
Yeah, these virtual seminars sometimes don't always lend themselves to organic sort of discussion, but let's see.
I have a question for you.
Hi.
So my name's Adrienne Randolph, and I am doing a big study in children, looking at both acute COVID-19 and multisystem inflammatory syndrome. And I was wondering, to me, it seems like a spectrum with this inflammatory response where, some patients are getting better and then all of a sudden they get cardiovascular involvement and multi-organ involvement. And you talked a bit about that, but you didn't talk as much about phenotyping these syndromes, both with laboratory as well as just clinically, because in the adults, they seem to all come in with respiratory, and then they get later on, even when they're getting better, then they'll recrudesce and get multi-organ involvement. And the children basically don't have their lungs as much involved, but they have the inflammatory involvement and a lot of cardiovascular involvement. So I was just wondering what your thoughts were on that sort of spectrum.
Yeah, no, it's a very good question. And so I'll say a couple of things. So I mentioned that we had collected nasal swabs. One of the motivations for collecting the nasal swabs was to see whether we could profile immune responses together with clinical data to identify markers of disease, immunological markers of disease or disease progression. Now we've now, our cohort is roughly 4,000 patients or 4,000 samples. Of those, I think we have maybe, I don't know, 400, 500, 600 positive patients. And that we've sequenced, genotyped, and what we've done is we've used this to build classifiers using the transcriptional signatures to build classifiers that are associated with outcome. And the outcome can be mortality, morbidity, severe disease, people who stay in the hospital for a long time, those who come in and then go home and are totally fine. And I'll tell you something that I found really remarkable. First of all, it works, meaning that we can find signatures that are associated with severity of disease. And these signatures are often weeks or even months before patient outcome. The amazing thing is that for children, they often have this very same, otherwise pathogenic signature, but they do totally fine. They appear to tolerate what would otherwise be a pathogenic signature much better than, those who are otherwise healthy and older. I know that this is not particularly satisfying and to directly to your answer, but it's to say that it's clear that there are some set of physiological features that allow children to tolerate infection better than their adult and older counterparts. Why some children don't tolerate it, I think, is an open question, and I think is going to be particularly important to answer, especially now that we're seeing many of the children we're saying that they do just fine. In fact, develop one syndrome or another weeks or months after they've cleared infection. So it's very much sort of an area that we're actively trying to explore, but I imagine there are others that might do a better job of profiling those kids better than us. Does that help sort of answer?
I think that's I think that's very interesting. Do you have a spectrum of age going down to these young adults that are you seeing age as a big, do you understand why age is such a big outcome modifier?
Do we understand why? We believe we find signatures are associated with sort of poor outcome. Why a young person tolerates that signature, that otherwise pathogenic response, more than an adult is not clear. Again, I know that's not particularly satisfying, but there are clearly patients that one is young and one is old. They have virtually identical responses. The young will do just fine and will tolerate that, and the old will do poorly. And this is it's not a novel concept. Of course, the immune tolerance, and I don't mean immune tolerance in terms of antigenic tolerance, but immune tolerance to pathology is a concept that is many decades old, and I think there may be some component of that in COVID. It's a matter of now trying to understand really what the underlying biology is.
Mm-hmm. No, that's very helpful. I mean, the same is true of all sepsis and severe infections. Is that kids very rarely die compared to adults. That they seem to tolerate even severe sepsis and septic shock as well, and with a fairly low mortality.
Yeah, there's some interesting genetics that's associated with this as well. But that's something that we're still sort of teasing out. But certainly that locus on chromosome 3 appears to be, at least in part, associated with some of these sort of signatures that we're finding.
Well, thank you.
Yeah. I'm sorry I couldn't be give you a more satisfying.
Hi, Sagi. This is Asimenya.
Hi there.
Thanks for your talk. I think your platform is really very suitable to study co-infection potentially of other viruses with SARS-CoV-2 and maybe the effects on SARS severity or COVID severity. So have you looked at that, or are you planning to look at that?
We have. And there are there are a couple. So yes, we have. We do find both changes in sort of microbiome profiles in severe versus less severe infections, and part of that microbiome is bacteria, and part of it is viruses. And there are certainly infections that are associated with severe disease. There's a very strong association, or we find a very strong association with herpes and other sort of otherwise latent viruses. The problem, I think, with that is that, intubation, so patients that are intubated and are infected with SARS-CoV-2 have a much, much higher rate of herpes reactivation. The problem with that is that I believe that mechanical stimulation in the lung is probably sufficient to reactivate some of these infections. And so it's hard to understand whether reactivation is making the disease worse, or it's simply a sort of byproduct of severe disease being intubated. I think what may be interesting is to see whether some antivirals may help in some of these sort of severe cases. As I'm sure you know better than I do, the trials with remdesivir simply weren't that satisfying, but perhaps targeting antivirals to patients that have reactivation of some of these latent infections, maybe that helps those patients more. I'm happy to share some of this data with, again, I should have said this at the onset. The data that we've generated, we're now writing up, but I'm happy to share data with folks before any of these publications or anything going online. So if you think any of it would be helpful, feel free to reach out, and I'm sure we could find a way to share data.
Hi, I'm Amar Majmundar. I had a quick question from nephrology here. I just had a quick question with your analysis in the UK Biobank. When you looked at variants that were correlated with, I suppose it was COVID-19 severity, were those common variants, or did you look at rare loss of functional alleles? What
They were common variants.
Okay, in those cases. And did you look at expression QTLs, or
we looked at expression QTLs, and in fact we found a couple that were quite very strongly associated.
I see. Okay. All right. And so, and have those been just in terms of a validation set? Were you able to validate that in an independent data set, or were those just two independent data sets within the UK Biobank?
Those are two independent data sets within UK Biobank. Those alleles do replicate in the much larger cohorts. The problem with GWAS in infectious disease is, and I don't know who's in the audience and who I'm going to offend, but they just don't work for infectious disease. And there are just too many places where the immune response can break down. So it's hard to find something that's going to be genome-wide significant. Yeah. But if you have a targeted set of factors that you're interested in, I think it's much more likely to find something interesting. Of course, you then have to prove it and so on, but there's also long literature showing that there's some involvement of complement and coagulation in COVID. So it's not that far of a stretch, but now when we're combining genetics with transcriptional sort of profiling, they fit quite well.
And then I guess the other question I had that helps me understand, because those are at least potentially host factors, even if they're difficult to address, that could correlate with disease. But in terms of pathogenic factors, have you looked into the evolving data of COVID-19 variants and are any of these new variants that are evolving creating mutations or disrupting potential mimicking proteins that you described in the earlier aspects of your talk? Like, are any of those proteins becoming mutated within new variants of COVID-19 and affecting pathogenicity?
Short answer is we don't know. Longer answer is, first of all, if I didn't point out, I should have, that, complement function is also associated with diabetes. It's associated with BMI, high BMI. And it may be that some of these patients are simply primed, physiologically, they're primed to sort of develop these coagulopathies and complement-mediated pathologies, and that's why. And there is, in fact, and this has been known for a very, very long time, that with age, complement activity sort of goes up. Some of it may have to do with that, or it may simply be a red herring. With respect to the mutations, although there have been, I don't know how many, what we're up to now, like 300 genomes that have been deposited. I'm sorry, 300,000 genomes that have been SARS-CoV-2 genomes. They're not complete genomes. People have been sequencing the spike. And so we actually have very little information about how the rest of the genome is evolving. The mimics that I showed, they're interesting, but I want to just say that mimicry doesn't necessarily, structural mimicry doesn't necessarily mean functional mimicry, of course. And so we don't know to what extent that mimicry or sequence evolution changes the functional properties of the virally encoded proteins. Having said that, we do have evidence now that some of these mimics may be inducing autoantibodies. Because of course, antibody binding is a combination of affinity and avidity. And so there may be sort of structural determinants that induce, sequence determinants that induce an antibody response, but then structural determinants that allow for cross binding of antibody. And some of that data is sort of now beginning to develop, and there are groups in Europe that are looking quite closely on that.
Thank you very much.
Yeah. And if you or anyone else has data about that may help sort of answer some of those questions, we're happy to help look at it. Or
any other question from the audience? If not, thank you so much, Dr. Sagi, and also Elle for co-hosting this meeting. All right.
All right. Thank you so much. And if anyone has other questions, you can feel free to send an email and reach out.
All right.
Thanks very much.
Thank you. Have a great day.
Take care.
Bye.